So definitely my pleasure. I'm actually turning into the Department of Biology. My pleasure to introduce Tarika Herman, she goes by T. She was an undergrad student in the genetics course-based undergraduate research experience that I teach. And so she's done this one semester project in that class and wanted definitely to present it here at the JSU Student Symposium. So without further ado, she's going to tell you about her project and using the nematode model to study hearing loss.
Great. Hello. As Dr. Perry said, I am Tarika Herman, or just T. I took the gene cure class with Dr. Turner last semester. I'm going to start off with a little bit of background. The overall goal was to assess the pathogenicity of a clinically relevant missense variant of uncertain significance using C. elegans as the model organism. A missense or a variant of uncertain significance means that there is a patient with a condition and a mutation has been found in their DNA, but it's not sure whether that mutation is causing that condition or not. So that's what I was exploring a little bit further. The next question I normally get is, why would you use a worm to study human disease? First of all, 83% of nematode genes have a human gene ortholog. So if there's a human gene you want to study, there's a very good chance that you're going to find a nematode ortholog for that. Additionally, they're small. You can store a lot of them. They reproduce quickly. From embryo to adult is approximately 3 days. So you can go through multiple phases of experimentation and build your data set very fast. And they're also transparent, so any type of abnormalities internally or externally are fairly easy to see under just a microscope. So I chose to study autosomal recessive non-syndromic hearing loss. Non-syndromic meaning this condition is not accompanied by any other signs or symptoms. It is the most common type of genetically inherited hearing loss, accounting for 80% of cases worldwide. It is typically sensorineural, meaning it is characterized by abnormalities of the inner ear. It is usually severe to profound hearing loss with a prelingual onset and non-progressive throughout the lifespan. There is no cure for hearing loss, and treatment is largely supportive, consisting of traditional hearing aids, cochlear implants, or the use of alternative forms of communication like American Sign Language. The human gene I chose to study in connection to this condition is MY06, which encodes for myosin motor protein 6, or just myosin 6 for short. To the left, you can see a structure model of what myosin 6 looks like. It is the only myosin motor that walks towards the minus end of actin filaments, which gives it a wide range of responsibilities in the cell, including vesicle and organelle transport, and it is essential to maintaining the structural integrity of cells. And I'm focusing on its involvement in the inner ear hair cells. It is powered by ATP hydrolysis, and it walks in a hand-over-hand motion. Mutations to myosin 6 have been shown to negatively impact the stereocilia of the inner ear by fusing them together. The stereocilia of the inner ear are small finger-like projections on the inner ear hair cells that bend in response to sound waves, and the bending produces mechanical energy that is transferred to electrical energy and sent to the central nervous system via the auditory nerve to be translated as sound. So fusing them together prevents them from bending, prevents them from transmitting signal, and so you have hearing loss. The 3 pictures at the top of the screen, the first 2 show normal, healthy stereocilia, and the third show mutated stereocilia, what that looks like. The nematode ortholog for this is SPE15, again, encoding unconventional myosin 6. It also has a wide range of responsibilities. In nematode, my focus is on its function in spermatogenesis, which is the production of sperm cells. Specifically, it is essential to the last phase of this process called spermneogenesis, where the spermatids are transformed into fully functional sperm cells. And mutations to this gene have been shown to result in an increase in unfertilized eggs laid by the nematodes due to dysfunctional sperm. SPE15 shares a 49.22% sequence identity with human myosin 6, making it an excellent ortholog to use for this project. Putting all of this together, my hypothesis states that introducing the human myosin 6 variant into SPE15 will result in an increase in unfertilized eggs laid by the homozygous mutant nematodes when compared to the wild-type N2 nematodes. Using ClinVar, I found my human variant. As you can see on the screen, it occurs at the 178th base pair, and it changes the 60th amino acid. At the nucleotide level, it is a change of a transversion from a purine to a pyrimidine, which is considered more severe because it is a chemical class change. At the amino acid level, it changes the glutamic acid to a glutamine, which is a amino acid class change of polar negative acidic to polar hydrophilic. This is important because changing chemical groups and changing amino acid classes gives the variant an extra potential to negatively impact the protein product and its function and structure. This does not prove that the variant is pathogenic, but it does give support in that general direction. Next, I completed a multiple sequence alignment across 6 different species to assess the evolutionary conservation of the variant location. On the screen, it is showing the amino acid level, and it is highly conserved in 5 of the 6 species. Higher conservation of a variant location suggests that that location is functionally important to the protein product, and therefore changing it could result in some negative impacts. Moving forward, a DNA extraction was carried out using nematode lysis solution. Approximately 20 to 25 nematodes were transferred to the solution, which was then put in a negative 80°C freezer overnight to assist in the freeze crack step, which essentially bursts open the cell membranes to liberate the DNA. And then it was run in a thermal cycler to assist in purifying the DNA sample and cleaning up any cellular debris surrounding the DNA, which, like I said, results in a purified DNA sample to be used for PCR. For PCR, I used Benchling to create my own primers. I created 5 sets, and the one you see at the top of the screen is the set that I chose to use. It has the lowest penalty score. And this was done to amplify the variant gene region for use in further experimentation. And then a gel electrophoresis was carried out to assess the success of the PCR in amplifying the variant region. You can see an illustration of what that process looks like on the screen, but it was set to 150 volts for approximately 45 minutes to allow the DNA fragments ample time to travel through the gel. And in the middle, you can see the scan of the results of the gel. Lane 1-1 shows my DNA sample, lane 1-2 is Dr. Turner's positive control, and lane 1-3 is the no template control. The target size for this was approximately 760 base pairs, and the largest band on the lanes were about 650, so not very far off, but could definitely use some refining there. I do plan on pushing this project further in Dr. Turner's research lab at the Wormhole this coming fall. So far, my results do support my hypothesis, and warrant further in vivo experimentation. My initial steps will be to optimize my primers and repeat my gel electrophoresis, and I hope to eventually be able to implement this variant into a live nematode model to assess the impact using CRISPR-Cas9 editing system. And those are my references. And I would like to say thank you to Dr. Turner and the JSU Department of Biology for their support and guidance through this process. Thank you.
Questions for our speaker?
Yes.
So I was just noticing that the gene you ended up choosing in the nematode is kind of unrelated to the one that's functional, unrelated in the sense of function. Are there other members of that gene family that might also be of interest so that you could sort of expand it out and look at others in nematodes?
There are a couple other orthologs. There is another SPE, I can't remember the exact name of it right now. But this one that I chose had the highest sequence identity, which is why I decided to move forward with it. And of course, you know, nematodes don't have ears, so its function was mainly in the production of sperm cells, so it's still a pretty good comparison for what it is, and the sequence identity definitely helps with that.